Okay, hey guys, how you doing? So we are going to look at taxonomy in this particular session. Now, taxonomy is one of those sections where there's a lot of kind of specific examples and stuff like that, and the majority of this chapter looks mainly at how we organize and classify different groups based on their characteristics and the main method in which scientists use. And so the key thing here is that I don't need you to remember sort of every single category and where each animal fits into where, but rather understanding the principle in which we organize it and the structure that we actually use to organize it. Okay, so let's get straight into it. Taxonomy. So here is a table showing all of the different number of species that fit underneath their kind of major categories. Yeah, so this is some of the things you are familiar with. We have about 386 species of mammals. And of those mammals, there's about 2 species of monotremes and 140 species of marsupials, and the rest are being placental mammals. 800 or so birds, 900 reptiles, and so forth. You can see of the animals there, the most numerous is actually the insects, yeah, with the beetles composing of the biggest category, yeah. So that's just a few things. There's a few other things that you might or might not recognize. Crustaceans, anything that kind of is segmented in terms of its body structure and lives in water. We have about 7,000 species. This particular one is called echinoderms. Those are your sea stars and your sea urchins, and then you've got other things as well. Yeah. Of the plants, we have about 18,000 species of flowering plants, and 11,000 species of fungi, and so forth and so forth. Okay. Now the key question here is, how did biologists sort of come up with that structure? How do you know or differentiate between a bird, a reptile, a snake, and a lizard, and so forth? And we have sort of ways in which we do that, which combines, the kind of classic definition based on structure, the biological definition, and also using genetics as well to identify and differentiate between them. Okay. Now, going through the definition, taxonomy is actually the study of identifying, naming, and classifying the organism. It's how we actually arrange organisms into the way that they are sort of arranged into, and it comes from the word taxon, meaning a biological rank or category. Yeah, so any sort of grouping method that we use for organisms is a taxon, and that includes sort of the ways in which we identify species, genus, families, and stuff like that. Okay, and this is sort of where it comes from. The main one that we use is this one here, which starts from life, and each time you go down, that is one particular group. So there will be from life you got domain, kingdom, phylum, class, order, family, genus, and species. And so what you can have is, if you look at this particular example here, you can have multiple species, or in this case multiple genuses, fitting into one family, and then multiple families fitting into one order, and then you would have multiple orders fitting into one class, and you sort of work your way up until you kind of get into the category of life, really. So let's go with an example. So here you've got kingdom Animalia, working down phylum Chordata, meaning chordate, or a organism that has a spinal cord, which is what we fit under because we go under the class of Mammalia or mammals, and this is where we break off, right? The order of Carnivora, meaning exclusive carnivorans or only eating meat, and then within the order of Carnivora there are 3 major families: the Felidae, the Canidae, and the Ursidae. Yeah, so Felidae meaning for all your cats, Canidae for your dogs, and Ursidae meaning the bears. And you can sort of see there the different genuses for Felis and Panthera. Felis for kind of your small cat, and then Panthera for your large cats. And then of that you've got Felis catus or Felis padalis. The padalis is the your lynx and your kind of medium-sized cats, and then Panthera perdis here, which is the leopard. Yeah, and so that's the kind of the different groups of cats. But you can sort of see that it's like a branching of a tree, really, that it kind of branches out, major groups branching out into smaller groups, and then until you get to the species. Now that's the kind of main structure that we actually use for classifying all of life, and you'll see there that there's kind of different ways to go about it, but this is the kind of generic method that we use. Okay, now let's look specifically at animals. Now I won't go into too much details, but let's see if we can try and find and track down where specifically some of the animals that we are kind of really commonly known and where they sort of fit. Yeah, this is how we actually organize it. Now the little black marker there is the critical innervation. That is the main structure which separates one branch from the other. Okay, so the main structure you see there in animals is multicellularity. The fact that we have multiple cells that work together defines our particular kingdom. Now, of that, if they have specialized tissues, we sort of break them off into this group, and you can see their sponges don't have specialized tissue, the periphery, and all their tissue are exactly identical, which means they don't have a brain tissue and they don't have heart tissue and they don't have any of those things. They've just got their kind of spongy tissue, and that tissue sort of just functions as it does. If it has bilateral symmetry, meaning if it's cut down the middle and the left side is identical to the right side, it sort of breaks off from there, and you can sort of see there jellyfish and corals don't have bilateral symmetry, which is why they fit under that category. And then as you sort of move through, having a body cavity, you don't actually realize it, but your heart, your lungs, all your major internal organs sit within a cavity in your body, right? And that means there's this kind of this gap in your body that allows you to store all of those major organs. You've got animals that actually don't have those major cavities, and so something that doesn't have one is, for example, a flatworm. They don't have a major body cavity to store all of their organs and things like that. Yeah, and then you've got a thing called a coelom. Now, I won't go into too much detail what a coelom is, but it's a bit of an extension of what that cavity looks like, and whether or not it has a kind of circular formation or segmented formation. Yeah. And then you've got other things like having a development of an endoskeleton, meaning that the skeleton is actually on the inside and not the outside. And that's what actually separates the majority of animals like chordates and things like that from your insects and your arthropods. Yeah, they don't have an endoskeleton. Yeah, mollusks have a shell. Worms don't have anything. They've just got a hydrostatic skeleton made of water or fluid. And then arthropods have an external shell, which is a suit of armor that they have to continually shed every time. Yeah, an exoskeleton. Now we fit under this particular category, the chordata category. Yeah, which is meaning having a spinochord or a notochord, and your sea stars are actually our closest relatives in terms of major groupings. Yeah, and they don't actually have those, but they do have a really well developed endoskeleton. Okay. If we go a little bit further, this is not a very good graph, but you can sort of see there. If you go from the chordates, you can sort of make your way through, and you can sort of begin to see the formation of different organisms reaching eventually to the mammals. Right? You got things like tunicates, sharks, and their relatives all sort of fit under this category here. You've got your bony fish, which is your kind of ray fin fish that you and I are all familiar with, and then you've kind of got frogs, toads, amphibians, breaking off into tortoises, lizards, crocodilians, birds, and then eventually your mammals. And so each one having a different feature: vertebrates, jaws, true bones, lungs, four limbs, an amniotic egg, which is a kind of an external egg, and then being endotherms or warm-blooded. Yeah, and so those are kind of the main categories that which we differentiate. We fit under this category here of mammals, but birds are also endothermic as well. Going one step further, now we get down towards the order. Yeah, so the order, in the order level, you can sort of see that you now begin to look at the different orders of mammals that we have. Bats, horses, and stuff like that all have kind of their own names, and humans and monkeys fit under the category of primates. But you've got other things, rodentia, meaning for rodents. All major rodents fit under the category. And then you've got a few other ones as well, things that you and I are somewhat familiar with. Here we are. Your carnivorans are here. Cetaceans, meaning whales and things like that, and cows fit under this kind of category here. Rodents are there. And then if you look at the order of carnivorans, once again, you've got your major categories again. Your felines, your bears, your canines, and you can see a few other there as well. Hyenidae, meaning hyenas, and then you got a few others such as your seals and things like that as well. Yeah, and so that kind of shows you quite well how we we categorize, and each of these things we use kind of structural features to determine those things, and so yeah, and that's sort of how we go about it. Now, in taxonomy, one of the greatest difficulties we often encounter is that how do we actually differentiate between lineages, which is, which group actually came first, which group came second. And so that method is actually what we call cladistics, of how we group them according to the characteristics and how we sort of arrange those characteristics. Yeah, and there's sort of two ways we go about it. We can either go by primitive characteristics, which seems to be the easiest thing that we can do, or we can use derived characteristics, which is kind of your more advanced features that appear in some members of groups. And so, we understand that all members of a particular group, for example, mammals, mainly, all mammals have 4 limbs. That's a primitive characteristic that is common in all mammals, and their ancestors show those characteristics. But then we also have to understand that there are kind of advanced, more kind of features that sort of come out from a particular group. For example, carnivorous mammals having claws is a derived characteristic. Yeah, and so there's different ways that we do that, and we use that information to then organize, okay, which animal came when, and that's how we sort of know how animals are related to one another. So I'll give you an example. So here are 2 different ways of going about it. Now, we're not saying that neither one of these are correct or incorrect, but there is kind of evidence to support one and there's evidence to support the other. And traditionally, this is sort of how we've always classified animals, right? In particularly your mammals, right? And so mammals and so your vertebrates. We've sort of always classified all mammals in one of their own category, and then they sort of broke off early on in their own, and then we've also classified that birds broke off on their own as well. And then in the middle, you've got your kind of reptiles, right? However, from a cladistic point of view, if you look particularly at primitive features and then derived features, if we arrange them in that particular order, the mammals are actually said to have broken off first, and then you've got your turtles, lizards and snakes, and then your crocodiles are actually last. And birds are really just a glorified reptile, a reptile that can fly, really, if you look at their particular features, because they exhibit the same features as other reptiles, except it's just more developed. Yeah, so instead of scales, they actually have feathers, and feathers are more of a representation of scales than they are of hair, which is what mammals have. And so depending on which way you go about it, if you go about from a traditional sense, then you might follow this method. However, if you look at their animal features and you classify them according to that, you might get a different set of arrangements. So in this particular case, you might say that birds are actually more related to crocodilians than they are to snakes and lizards. However, in this particular section, you might say that they are a little bit more closely related to the snakes and lizards than to the mammals or something like that. Yeah, and so that's just different ways that we classify them. Okay, let's talk about kingdoms. And so kingdoms is a kind of the first kind of segment below domain that we looked at. And there was actually, for a while, they couldn't figure out how many different kingdoms there were. And Linnaeus, as we talked about last time, the godfather of biology, developed 2 major kingdoms back in the 1700s. He basically, everything was either an animal or a plant, and that's just a kind of portrait of what he looks like. And then a guy named Haeckel in 1866 actually added a 3rd category, and he didn't really have a particular name for it, or he did, but it didn't really have any particular meaning. But he knew that there were some things that didn't fit neatly into animals and plants, so he developed 3 kingdoms, which is the animals, plants, and everything else category. Once again, not exactly the most brilliant and specific way, but it was the best thing we had at the time. And then a guy named Whitaker actually developed a more specific category. So he developed 5 kingdoms, which is actually still the most prominent method that we use today in 1969. Animals are 1 kingdom, plants fit in their own kingdom. He had a kingdom for fungi, and then he also had a kingdom for protists, which is kind of your separate category. Some of the algae will fit into this category, some of the flatworms, some of the kind of single-celled bacteria and those kind of organisms will fit into this category. And then he's also got your kind of Monera or prokaryotes will fit into that category as well. Right? And a guy named Weiss then derived another sort of method, which is looking at your 3 domains. So he took the bacteria out of the prokaryotes category, and then he sort of gave them their own domain. And then he had another category and then you put eukarya, which is eukaryotes, all in the same category, right? And so once again, you kind of get these different methods of classifying. Nowadays, we actually use a combination of Weiss's one and Whitaker's one, even though technically Whitaker's one's a bit outdated. Okay, let me show you diagrammatically what it looks like. Animals, all the major animals we just talked about are there. The fungi, your kind of mushrooms and your molds and your toadstools fit there. Plants go in there, and then some algae species fit under the kind of plant category, some of them don't. Some of them fit under the protist category. Amoebas, yellow algae, and so forth fit under these. And these are kind of like your eukaryotes, but they're your single-celled eukaryotes. Yeah, and then you've got your prokaryotes, which is your bacteria and all those other ones as well. Okay, so that's the 5 kingdom classification that we use nowadays, and we still teach this quite often, like you guys are learning it right now, but it's actually quite outdated because technically bacteria occupy their own domain, not their own kingdom. Okay, and that's sort of what it looks like. Yeah, and then you've got characteristics of each one, right? And I won't go into too much detail, but the main thing here you can sort of see there. Unicellular are used for Monera. Protists can be uni or multicellular, same with fungi, but your plants and your animals are multicellular. Difference between animals and plants is actually the cell wall, with plants having a cell wall and animals not having a cell wall. Photosynthesis is one of the main differences as well, being autotrophic and animals being heterotrophic. Yeah, and you can sort of see major differences shown there. Okay, all of the species are eukaryotic with the exception of the Monerans or the bacteria, and they include things like cyanobacteria and so forth. Now this is Weiss's kind of 3-domain category, yeah, and it kind of gives you a nice perspective on sort of how big or how many different categories there are. All the animals fit into this particular category, right? And so you can sort of see there that we're just really one branch of many different branches, yeah, and you've got a whole host of different bacteria and archaea, which is a kind of primitive bacteria as occupying their own domain, right? Now, there's really a lot more to be discovered. So when we talk about species and stuff like that, we're discovering new species all the time, and roughly about 17,000 new species are being discovered every year. Yeah, a few examples. In 2004, they found a new species of small marsupial. So the black-tailed Antechinus was discovered somewhere in the altitudes, high altitudes of New South Wales and Queensland border. So that was a new species that they discovered recently. In 2014, they found a new species of sea anemone that actually hangs upside down on ice. So that was pretty cool. Like, you kind of get these random discoveries that they have never sort of picked up before, but you can sort of see they're just sticking on the upside down parts of ice. It's pretty sick. And then they found a translucent glass frog that was found in the wet tropics in Costa Rica in 2015. So that was pretty cool. You can see there, you can actually see its internal organs there as a result of that. Yeah, so that was pretty cool. So yeah, that just gives us a good idea of some of the different organisms that we use. Nowadays, we're pretty good at tagging, identifying species, and then immediately categorizing them. We use a quick ID method. Traditionally, we would have to look at the structure and break it up, but now we use a particular thing called a DNA barcode, and it's pretty convenient because it's 650 bases in the gene, the CO1 gene, that's present in the mitochondria. And so what we actually do is the DNA barcode helps us identify that particular organism, right? And so here is the boarfish's CO1 gene, and what we actually do is we look for similarities and differences between the barcodes, and the more similar the barcodes are, we can then match it together with all these other fish, and we can find out exactly which category the boarfish will fit under. And so you might get a few differences if it was a different fish of the same category, but then if you matched it against a reptile, for example, you might get major differences. And so that's how we use those minor differences in these barcodes that allows us to then ID the organism. Yeah, so that's a bit of an overview on taxonomy. I hope you guys enjoyed that, and I'll catch you guys in the next session. All right, see ya.